before if it was always that way when you've been annotating.
I think it's always that way, and that's just
Yeah, now I'm
because
just
when you're
getting
looking at
dragged.
the
Kim,
video.
you want to come up a little bit in the water column and then we'll reacquire.
When you're
Ship's
looking at the
currently
video
stopped,
on.
correct? Okay. Roger that.
When you're looking at the video on even a high
Alright, I
definition
think I have
monitor,
enough
10
play
centimeters
now.
is
Let
about
me
the.
see if I can turn and find you. Nope, negative. No, we
10
have
centimeters
a
is
good
about.
delta now. I'm just trying to back up and turn around to spin you to find you. Alright. See if that was good. Sorry, science onshore. We might be about another 10 minutes or so. 5-10.
Good. No worries. We're trying to entertain the public who's watching, but also let you chat as you need to chat to get situated.
Sure thing.
Yep. So the 10 centimeter rule is mostly that's the size at which you can really see what a species is. But beyond that, the IDs get really tenuous.
That makes sense.
Oh. Would you mind trying to spin around to find me there? Seems like I don't have a tether for it right now. Sure thing. Sure thing.
As we've been up off the bottom, there have been a couple of little lanternfish that I've seen skipping in and out of our view. They're very common deep sea species. They serve as a major kind of basal link in the food chain, and we saw one Pacific black dragon.
Yeah, I see us orthogonal to each other, or completely kind of 180 to each other. It's heading. Great. That's very helpful. Thank you. Awesome. Nice to be back. All right. So now so Kim, I guess we need to go back the 180 that you came, and then that's on the ship's heading, and then we can go down from there. Yeah. Yeah, sounds good.
All right, science, I think we're mostly on the way to sort it out. I would think you'd probably want to start that over. Correct me if I'm wrong.
We can start it over, yeah.
Okay.
I think I'm getting tugged again. Roger that. I should be coming under you now. Great. I see myself in your down cam, so we're Gucci now, guys. We're flying.
So there was a question, Jess, if you're comfortable answering it, about latency when you give commands to the ROV, for example, to fly or to move the arm.
Sure, yeah. So there's so because we're communicating basically at the speed of light, so we have a fiber optic cable, which means that we can communicate pretty quickly, you know, speed of light. You really don't experience a latency when we're flying 13 delta there. Yeah, I'm going down. Yeah, so yeah, you don't really experience a latency when we fly. It's also my Z-bias is all wonky. Sorry, I'll go off SPL for the ops comms.
So there's also a timely question about how to manage bad weather during dives. We monitor it very carefully. So right now we actually have a big weather system moving in from the west, and that is really impacting the length of this dive. So we're planning to recover the vehicles in just a couple of hours so that we could have them safely aboard the ship. We need to keep an eye on wind and swell so that the team can get the ROVs on board without them hitting the ship or without anybody getting injured. So that's something that we monitor and talk with the ship's crew about to determine those types of things. And actually after that,
it makes sense, at least with the bathy. I've lost kind of a sense there. What are you facing? You're at...
After that, we actually are heading to an area of refuge for probably a couple of days because of the system that's moving through. So we'll do some mapping on our way north and get to someplace protected for a couple of days.
Yeah, the forecast out there right now is quite impressive.
There's a question about how the lights affect the life that we see on the seafloor, how sensitive species are to it, and whether it scares them away.
We do see some evidence that in some of the fish species, they find it confusing and disorienting. There's the species of eel pout that's frequently up in the water column, and one of the things you'll see is it will swim down into and often into the sediments when it encounters the lights. But we've also had fish, particularly rockfish and halibut and some of the lanternfish, that will use the lights of the vehicle as like a hunting strategy. So we've had this happen actually just south of here at AZ Bank with a different vehicle where we had rockfish just all over the vehicle because it had attracted krill, and they just followed it around. And in previous Nautilus seasons, we've seen like clouds of either the lanternfish or the some mackerel that we weren't seeing them often on the camera, but you could see them in Argus cam, and they were just a cloud that were kind of following it around. So different animals respond slightly differently. It doesn't really seem to bother too many species down here that much, but it's also ephemeral. So we're here and then we're gone, and they go back to whatever they were doing on the bottom before we got here.
I've also heard that the noise of an ROV can be very distracting for the life that can move, that can get out of the way. They don't really want to be associated with it, at least for some organisms. And the problem with that light aversion, if it exists, and that noise aversion is that my office is NOAA Fisheries Office of Habitat Conservation. We're really interested in the role that deep sea corals and sponges play as fish habitat. We work with fishery management councils who try to manage human activities that affect fish species. And if we need to protect deep sea corals to make more productive fish habitat, then fishery management councils are more than happy to do it. But we can't tell very easily whether corals and sponges, or to what extent corals and sponges are really acting as fish habitat if they are indeed affected by light and sound of the ROV. So it's an excellent question, and one we really do need to work more on. But it's good to hear, Meredith, that it's probably not that big of an issue.
Yeah, well, and we're starting to apply different tools to this as well. So that's one of the things we're looking at in eDNA is the fish communities that we see in addition to the corals and sponges, because even if the fish has moved out of view of the ROV, it will still have left a genetic signature. And so we can tell that it was there sometime in the relatively recent period preceding that. It also lets us just detect species we haven't seen. So as you can see watching this video, we can see whatever is in the light pool, but you can have a really cool fish or cool coral that's just behind you or just outside of the range of that camera and light, and you won't know it's there. You can often pick it up in environmental DNA.
Just for the scientists, we have restarted the transect now. So,
Okay.
yep, just to give you guys a heads up.
And we're
And have
still
we
calling
numbered
this
this as
10.
11 or still 10?
I'm not sure.
Guess you get to pick, Meredith.
They're asking if this is going to be number 11 or 10 still.
Oh, right. Is this still going to be 10, you guys?
Let's restart it as 11, and I have a note to pass on to Tom that 10 was aborted just due to mechanical issues.
Okay, sounds good. Thank you.
And what's the direction on transect 11?
055 at the moment.
Great, thank you.
Whoa,
That looks like,
that's
looks like a
cliffy.
cliff.
That's very cliffy.
Yeah.
There's not much.
Oh, wow.
So, okay, so to pull it along, I think we're going to have to move north, yeah?
Yeah, along this, yeah.
Yep.
Sorry, that was a bit of a surprise. Bridge nav.
So I'm changing the direction on the transect is just fine to just keep the screen
20
in view.
meter step due north. Well, there goes my theory about a gradual.
A nice little slope failure there. You could see all the layering. Stratigraphy.
Yeah.
If we weren't on a transect, I'd love to look at that, but
Or technically
Move
kind of
along.
pause mid-transect.
Not even mid transect, like just
10
I
seconds
just
in.
hit enter.
This is an interesting little cutout though.
Wow.
Yeah, it looks like a slice of cake.
Yep,
I like the little biology on the top that looks like it's a footprint, though.
it does.
Oh yeah. Who was here? Somebody.
Just one step back for the perfect photo.
So for the folks on shore, do you guys want to see kind of the strike of the wall this way, or would you rather see the flat bits that are kind of more off to the right?
I'm not sure what you mean. You mean like following the edge or following the top?
Yeah, exactly. So do you want us to follow kind of this steep edge, or do you want us to follow the kind of the sandy bottom here?
Let's follow just along the top steep edge there.
Okay, Roger. So something about here is what you want to have in mind?
Yep, that would be great.
Sure.
Yeah, often we find these kind of top edges are great habitat for things.
Kurt Whitmire has just entered in the chat room that NOAA Fisheries is just starting to wrap up an untrawlable habitat strategic initiative with a major objective of measuring the reaction of fish to various mobile survey platforms, getting at the light and potentially noise question, or maybe just light. So for those of you interested, if you follow along on NOAA Fisheries communication channels, that report may be coming out soon and should have lots of good information about different fish species attraction or repulsion from these things.
You wanted to know what depth we're at. We are at 917 meters depth right now. We started this dive at about 1,300 meters. So we're moving up the canyon, looking at the north-facing wall.
And we're still in this little box canyon feature. I think that they had planned into the dive report because there were some geology teams that were interested in it and whether or not it might have been a slump or something else. And I think we're getting them some good footage of what the canyon wall looks like in that feature.
Absolutely, and geology doesn't always get as much love as the biology, so it's really nice to be seeing interesting features like this.
Thorny head, fish on the left. I've got a couple little crinoid corals on that boulder at the top of the screen. It looks like there's a bit of current pushing up over this edge.
Yeah, there is actually. It's kind of pushing us into it.
You just see all the crinoid arms like waving in the undersea breeze.
A viewer wants to know about the little red fish that we see sticking to the seafloor.
Those are thorny heads, and they are actually in the rockfish family. They are the deepest rockfish that you will find. Most of these at this depth will be long spine thorny heads. Their common name is the idiot fish for the poor thorny head, although that's kind of an open question of whether or not that's because there's something about the thorny head itself or because their head is absolutely covered with spines. And so if you were to pull one up in a net and then grab it, it might say something about you.
Yeah, I'm voting against the latter. That's such a great name.
Video's changing out.
It's a good thorny head right there. Alfaria sponge just going along the bottom of the screen. Collected one of those on this dive.
So for viewers
Really?
on Nautilus Live, we're, and for actually our lead scientist ashore, we're just doing a dinner relief right now. So there's going to be a brief lull while we trade people out.
Excellent.
Meredith, you mentioned eDNA work that's up and coming right now and is in a really exciting place of discovery and potentially helping with lots of questions around fish habitat use for deep sea corals and sponges and plenty of other questions too. What's the most exciting thing right now in eDNA research? What do you go to the lab for in the morning?
I mean, for me, it's the biodiversity surveys. One of the nice things about pairing eDNA with the collections and the video and working in these deep sea habitats is it's iterative. So we find specimens, we can then look for them in environmental DNA, get a snapshot of the biodiversity, but we also will see the sequences, the barcode we use to identify species for species that we don't know yet. We can usually tell what family they're in, but they just come up as unknowns. So then we know that in that area, we have a species we've never seen before, or we don't have in our collection. It might be in a museum somewhere. So it gives us a new aspect to look for. So we can capture bigger biodiversity and compare it back to the video and identify things that we might not have been able to identify before. And so that I find that to be really exciting and fascinating. and just a way to get, we like to think of it as it's just a different type of net you can study the ocean with. And it's also nice when you get into these big gardens where you can't pick up every single animal, but you can tell an overall picture of who all is there without having to, using this technique.
Mm-hmm. And how long has it been a technique that you can use? Like when were the first eDNA samples of the deep sea taken?
We took our first samples with Nautilus actually in 2016 in their Cascadia margins. And I think we were some of the first for the deep sea stuff. I think Ambari was doing a little, but we were very early.
Mm-hmm. Until 2016, that's only 4 years. That's amazing.
Mm-hmm. That was another cruise that was seeps and coral.
Mm-hmm.
Sounds of home. Someone has a dog that's Heather.
Sounds like Satchma. No, my dog does not look like that. I think that's Meredith's.
That is mine.
So there's a question about backgrounds and what it takes to become a deep sea explorer. Does everyone need a degree in microbiology, or are there lots of opportunities out there? So I think anyone can answer that question, but maybe we'll start with Heather. Talk a little bit about your background.
Sure.
What led you to ocean science?
Yeah, real quick, going way back when I was about 5 years old, my older brother, who was studying marine biology in college at the time, would bring me tide pooling. Yes. He would bring me tide pooling, and I just fell in love with the ecosystem completely. The fact that you could, that there were animals under the waves that you couldn't see just blew my 5-year-old mind, and it never stopped. So, so I followed in his footsteps a bit. Studied marine biology and marine ecology in Southern California, where I'm from, and then continued after that in British Columbia a little bit. Then I moved from research, which I had dearly loved, but I'd always wanted to get more into the policy side of how do you actually implement research in a management sense. So I started working at the National Academy of Sciences to coordinate teams of incredibly smart people who have a lot to say, but maybe aren't the best communicators. So we would we would gather the message that they would all be telling together and turn that into policy recommendations. And I loved that job. It was a lot of fun to do that, but it was a short-lived postdoc. I was fortunate after that to end up at NOAA, where I am right now. I think that was about 4 years ago, and started working at the Deep Sea Coral Research Technology Program, and that was the first time I'd done real deep sea work. I had kind of dabbled a little bit in grad school, but this was dedicated. Let's figure out what's going on the deep sea seafloor, and how we can protect these resources that are so valuable for our fisheries and for their own sake as well. So I guess for me, it was really studying a lot about marine ecology, how these animals work, essentially not as much the physiology, but how they respond to environmental conditions and how they act as a community to work together and use habitat, so fish using habitat like deep sea corals and sponges. It was that plus studying the social side. So I learned some economics on the way, and that was critical in understanding what people care about and why, and how we can use what people care about to protect the animals that, and not only animals, but protect the whole seafloor that is supporting fish and other species. So that's a little bit about my background. Meredith has a very different background since she's still in the lab and doing this fascinating eDNA and other work.
Yeah, Meredith, how did you become the eDNA expert?
I actually, way back as an undergrad, started with a background in biological oceanography and got a degree in that from the University of Washington. And while I was an undergraduate, I had the opportunity to start working in a genetics lab that was doing phytoplankton genetics. And I got really into the genetics aspects of doing this type of research. So I went on to the University of Miami and got a degree there in the functional genomics of killifish. But the nice thing about using genetic techniques is that the techniques are the same, kind of no matter what you're working on. And so I've had the opportunity to work on a lot of different marine animal species, from phytoplankton to corals to whales. And I've been very, very interested in the deep sea corals lately, and for the last 8 years. And I think it's definitely become a major focus, long-term focus of mine, which is wonderful. And one of the big challenges we face is, how do you get samples? You're dealing with a very hard to access ecosystem and animals that are very fragile. And so eDNA offered us an opportunity to collect samples in a different way. And we're really trying to harness that a little more. We still need like physical samples to do the verifications and develop the background libraries we need to use the tool most effectively. But it's a really fun and really powerful way to investigate ecosystems. And so I'm happy to be involved with that. But yeah, it's just started with a long genetic background.
That's great. And there are a lot of diverse backgrounds in the team that we have on board. And I wonder if anyone else who's on watch right now wants to talk a little bit about their path to Nautilus.
Nicole, where are you sitting right now so I can?
I am in
SCF?
SCF, yeah.
Just want to turn you up a little.
I'll give a quick one. Just because mine's a little different from a lot of people who have science backgrounds, I have just a bachelor degree in journalism. And this is Marley, I'm a video engineer. And yeah, long story how I got to be working on Nautilus, but basically I started working with scientists, documenting research at a university, taking photos, filming videos. And if you work with enough marine scientists, eventually you get invited to work on ships. But yeah, I always try to encourage people who are in, humanities or other degrees that aren't specifically science. You can still find avenues to do some pretty adventurous, cool ocean work if you're interested in it.
Yeah, and I think that a lot of scientists are always looking for ways to collaborate with people who are outside of the sciences too. For example, Dr. Thurber, who's one of the chief scientists on this cruise, and my advisor, this is Leela talking in data logger position, has worked with Lily Simonson, who's a fantastic painter and who's also become really knowledgeable on deep sea organisms. But we were talking the other day about how they started working together, and she, basically was just very persistent and showed interest and eventually got invited out to do some really cool things with Andrew and also other awesome deep sea scientists. So yeah, just getting in touch with people really. Like you were saying, Marley, once you work with enough marine scientists, you'll find your way out on a ship, for example. Don't be afraid to reach out to people.
Leela, what happened to the urchins last night? We had someone asking what was done with the urchins and what is going to be
further done with them?
Yeah, further done with them.
Yeah, so those, was pretty simple. We just scooped them out, tried not to get stabbed, and we froze them whole. And we're hoping maybe to do some gut content analyses on them because we're interested. Those were collected from, I believe, near this, that was Seep Dive, yeah. Yep. We're just interested in if other fauna near those seeps use the seep-derived carbon. So if the seeps are providing a trophic subsidy somehow, I believe that was one of the things that we wanted to do with those urchins was gut content analyses. Yeah.
And And then another viewer wanted to know if we do live data logging or if video is reviewed, and in this case, both.
Do live data logging your what?
Or review the video after the fact.
Oh, yeah. Yeah, in this case, both. So I'm sitting up here, standing data logging watch, so I'm live data logging, and then I also go through and look at all the images afterward and pair them with the different samples that we've taken. And then also I personally will probably be maybe using AI. We'll see how we do it, but we'll be reviewing video footage to look, at least on the seep dives, at distribution of megafauna and trying to quantify that and maybe what the role of seeps is in providing habitat or trophic subsidies for those fauna. So that'll be some video analysis that I will likely do or be involved with.
And then similarly, Meredith, there's a review of these transects that we're conducting right now by the Deep Sea Coral and Research Program. Do you want to talk a little bit about that?
Sure. That work is being done with Tom Leidig's group from the Southwest Fisheries Science Center. He's been on and off the chat all day. And so we run these transects to be roughly the same no matter where they're done coast-wide. And so then they have someone sit down and review them, and they count every fish and invertebrate over 10 centimeters. So they use those lasers as the reference point. By doing this, and they do it in a kind of single track line just right down the center of the screen, so it's roughly the same kind of square footage footprint as the ROV travels along. That's why we're traveling at this slow, steady speed, about the same height over the seafloor. And by doing those counts this way, we're able to compare among locations up and down the coast and get a better handle on how the corals are using the habitat, how the habitat changes in different areas along the coast, and also just to provide the locations where a lot of these species are occurring.
An astute viewer noticed the holes in the seafloor and was wondering what created them.
You know, there are a variety of different invertebrates that can be burrowing in there, ranging from worms to small crabs, shrimp. So I'm actually not sure what's creating those burrows. Some of the sea pens we've been seeing can actually withdraw down into the sediment as well.
Yeah, so we're seeing a lot of crinoids and some Dover sole and thorny heads and occasional anemones.
Yep. Right, just past the lasers right now is a sea pen called Umbellula.
Yeah.
And coming into the lasers right now is a sea pen. We usually call this one a sea whip. This one is probably Funiculina, but that group at this depth, telling that from Stachytilum or Protoptilum can be very, very tricky.
I was catching the Umbellula. You mind throwing in the name of the one you just mentioned in the chat? So I don't butcher it. Thanks, Meredith.
Yeah, these sea pens here are also a good indication that there is no hard substrate for a ways down here. These are species that only grow in the mud.
Meredith, I know there's some back and forth around what we call aggregations of corals. You know, in some cases they're gardens, in some cases they're groves, and there's different definitions for different ones. There's so many crinoids that we're seeing. Is there a term for a group of crinoids?
The collective noun. We were looking up the collective noun for crabs earlier. Apparently it's a cast.
A cast of crabs?
Yeah, I'm curious what it is for crinoids, though.
I don't think there is a collective noun for crinoids, so maybe we can invent one.
Maybe we should.
Yeah.
I mean,
Googled
I'd
it,
say
now
captive
finding
crinoids.
one.
Mhm.
A meadow.
A meadow of
I like
crinoids.
it.
I like that.
That's good.
I have a really cheesy
There's,
one.
What is it, Erin?
yeah.
A shame. Like a crying shame. Shame
Oh,
of
wow.
crinoids.
Crying
Love
noise.
it. There was a fun trivia night we used to do in DC, and if you were in a tie with two teams at the end, and each team was in a tie for first place, the winning team had to guess the most correct names of groupings for animals. So murder of crows and all those
Yeah.
other tower of giraffes. So for a while I was pretty good at that, but I think I've
Yeah.
forgotten most of them.
I was looking up a list of those recently. There really just are the most ridiculous names. So, I mean, if there's sea lilies, we could also call it a bouquet. Bouquet
Oh,
of
love
That's another
crinoids.
good one.
A scattered
That would
bouquet.
be. That would do really well for the stalked ones too.
Right.
So someone wants to know how to follow along and watch dives after the fact. They scoured the internet and found the ONC dives posted at the CTube page that ONC has. If you go to Nautilus Live's YouTube page, you can actually watch back any of our dives in the last several years. So the dives from this expedition will normally be posted a couple days after they've actually aired live, but you can go back and explore parts of the West Coast that we've visited in previous years and yeah, catch up on what we've been doing. I would recommend catching up on the Monterey Bay expeditions since we're going back to that area this year, so you can kind of see a comparison of some sites, including things like the whale fall, where we discovered that last year and it was fairly intact. We're going to hopefully get another dive on it this year and see how that has changed. So go to Nautilus Live's YouTube page and check out the dive videos for that.
Science, quick question. So we're coming up on what would be a 200-meter transect in about 50 meters. That does include this bit of craziness going along that wall. Is that okay, or would you like us to extend beyond this point to get kind of 200 meters along this contour?
No, including the wall is just fine.
Okay.
So we can call it when we hit 200 meters here.
Okay, great. And so then when we finish that, point 1.12 is quite down slope. I don't know how important that is, or we can make our way to 1.13, whatever your preference is.
Let's stay up and go to 1.13 and just kind of contour around this feature here, just because we only have a couple hours left, and it would be nice to kind of get through this box canyon.
Sure. Okay, that sounds good. Thank you.
Someone wrote in to suggest a crackle of crinoids.
Crackle of crinoids. Yeah, the alliteration is good. They do that with area of eagles. Oh, that's not really alliteration. Oops.
A nice deep sea sole and another thorny head there. Some places, if you have the vertical walls, like the one we just passed a few minutes ago, sometimes you'll see the deep sea sole basically like suction cupped to this vertical wall. So I was like, how do you hang there?
Yeah, we saw that one earlier. It was crazy. Just hanging off the side.
Another animal grouping thought is school of fish. Been wondering if that's just fish that are up off the bottom, off the seafloor, or if demersal fish are considered schooling as well.
That I don't know. Although I'm betting it applies to all fish.
It's a school of thorny heads, just doesn't sound right. They're just sitting there.
Jenny in the science chat has just suggested a cram of crinoids.
Love it. They do cram.
Next, I'll have to come up with what is a group of sea pens.
There was a
plume.
viewer that wanted to know, she thought she heard you guys say that sea pens only grow in mud. They were wondering if that's true.
Well, it was true until it wasn't. There is now one known species that I'm aware of. It's the Anthoptylum, the big red striking sea pen that we've actually seen a couple on this dive, has one species. It's called Anthoptylum lithophyllum, and it's known as the rock pen. I think it was originally found in New Zealand and in Monterey Canyon, although it's been now seen up and down the West Coast in different locations. And sea pens have this physical structure that allows them to dig into the mud called a peduncle, and it's just the bottom end of their body structure. And in the rock pen, this peduncle has been modified into a suction cup, and they suction cup themselves to rocks, and so they only grow in rocks. And that's actually one of the species we'll be looking for Gary Williams throughout the season.
We about to call this T11E, Aaron.
T11E?
Is this about to be the end of it?
Oh yeah, it will be soon. Let's give it another, let's kind of finish out this 20
Yeah,
meters.
Let me know whenever you
When I
log
drop
it,
it,
and I'll do the same.
we'll do.
We must have a few pebbles mixed in here because we're seeing these Sola sea cucumbers again, and they've got to be glued to something.
Bridge nav, another step, same bearing.
And we've got one little Gersemia, probably growing on a small pebble, just going off the bottom of the screen, with that fluffy pink shape.
Wonder what that is popping up in there. Is that your tether?
Well, there's the Anthoptylum we
maybe.
were just describing is going off the lower left corner right now.
Data switching over.
So lead scientist, just so you guys know, the Aaron had said that we should plan to recover the vehicles at 7, so coming off the seafloor at 7 to have the vehicles secured in time.
Yep, I asked that actually just before you came in, and they told me 7, so
Okay.
we were prepared.
All right. Bunch of chatter going on as the dinner relief switches back over.
All right, we're into transects.
Excellent, thank you. And how, do we have any Niskin bottles left? I'm just curious.
Sorry, our data logger just is still plugging into comms, so she did not hear that question, but
no rush.
we'll relay it.
Hi.
Hey,
No, I was just curious what it was is all.
hey, Susie, do we have any Niskins left?
You put what?
Yeah, hold
No,
on
the
a
shiny
second.
thing. I was just curious what that was. No, I didn't change that
Oh,
at all. Sorry, the
Yeah, we
sector.
have 2 Niskins left, 5 and
Okay.
6.
Thank you.
Okay. So why don't we, if we can, once everyone is settled, before we start moving again, let's go ahead and trigger one right in here.
Jess, there's a request for a Niskin when you guys have a chance.
Roger that. I'm just getting set up, and then anywhere around here, the Niskin is good.
Yep, anywhere in here.
Sure thing. And is there a specific altitude you like, or?
This would be fine, or a little closer to the bottom if we can get a little closer down
Sure.
without stirring it up too bad.
Sure thing.
Some pool.
Meredith,
All right.
Meredith, you're looking for eDNA with these Niskin bottles, I'm guessing, and what else are you analyzing from the water samples?
So for what I'm looking for is just the environmental DNA. I think on this leg and possibly a couple of the others, there was some interest in collecting microplastics out of some of the water, just to see how much is in the area.
Yeah,
And
yeah, come up
on
on the
later
delta.
later legs, they will be analyzing for different food chain chemistry, so particulate organic matter and different isotope analysis.
And is that isotope analysis of phytoplankton or whatever else, whatever other kinds of plankton you're finding in the water, or what's getting analyzed?
That I am less
So do you
certain.
want to
I
look
think they're
down on
actually,
Argus too when you get a chance there? Thank you.
I think they're processing the water samples themselves and not necessarily the organics in them. So that would be they'll analyze the organics as just a component of the particulate organic matter.
Roger.
So I can chime in that we are also collecting the particulate organic matter for isotopic analysis for the people that are going to be doing that on later cruises as well. So that and the DNA are both coming out of the Niskins that you guys collect.
Yeah.
Awesome.
Excellent, thank you.
And it is more
Ellie,
for
full
the
wide there.
base of the food chain,
Thank
so
you.
it's things that are retained on a 0.7 micron filter. So that's mostly just detritus, as well as the very big bacteria up to the very small plankton, or phytoplankton kind of things. Not much phytoplankton at this depth, though.
Yeah, well, they're using a bigger filter than me. I'm using a 0.45.
Great, thank you.
All right, so just to
right.
clarify for operational stuff, we have Niskins number 5 and 6 available. So for this water sample, you can use number 5.
Great, thank you.
Sure.
And let's do that right now.
If there's not much phytoplankton making it down the steep, where does it tend to get eaten on the way down? Or does it not fall like other kinds of particulate matter?
Would you mind turn on the porch light there, please, Kim? And sorry for science, we're about 2.5 meters off the sea surface. Is that sufficient for you?
Yep, that's just fine.
Okay, great. Number 5.
So the phytoplankton that will fall all the way down, not that much of it makes it because it does get consumed on the way down. As we ascend, we'll probably see things like the larvation houses. And so they will filter some of that material out as well as other filter feeders like the salps. Some of it does make it all the way down, but definitely not what starts on the surface.
Yeah, I was always told it's somewhere between 1 and 10%, depending on the type of phytoplankton in the surface, make it into the deep sea. And the very deep sea, much less than that, even makes it down. And
You got eyes on the Niskins there?
Yes.
All right, 5 is fired.
All right, thank you. That is sample number 41.
Thank you.
And I should have been specific that phytoplankton does make it down this far, just there's not much light, so it's usually pretty dead at this point.
Yeah, it's forming some of these
Yeah.
fantastic marine snow clumps we are seeing.
Right. I did anticipate that it was dead, but I had no idea how much of it actually makes it down. Thank you.
One of my favorite statistics about that, which I have absolutely no idea if it was true, although it did come from a scientist, is that some phytoplankton, or by the time it makes it down to the abyssal depths, has been eaten by 7 different copepods from the surface all the way down. So it's quite a recycling chain on the way down.
I love that.
Okay, y'all. So we decided while you were at dinner that we're going to skip 1.12 because it's way down here. We're going to move towards 1.13 and try and hug in along this next canyon feature.
Sure thing.
Okay.
This is an exploratory mode, or are we doing another transect?
Exploratory, correct?
We are in we are in exploratory. We want to get at least roughly 200 meters from this last transect.
Great. Oh, we better get a move on then. Okay. All right, I'm going to just go 075. I think I'll do some big steps because it's pretty flat.
Sure thing.
Bridge nav. Can we have a 40 meter step bearing 075? Correct, 40, thank you. Yep, that's correct.
It's amazing how much smoother it is to move around when you're not dealing with canyon walls.
I'm still thinking about phytoplankton. Totally random question popped into my head. And I know that sometimes phytoplankton in your surface waters can be responsible for toxicity, red tides, things like that. Has anyone ever noticed any toxic phytoplankton issues not at the surface? I mean, no, they're not really reaching the deep sea necessarily, but where do we see those responses?
I've never heard of any impacting any kind of deeper like mesopelagic or mesophotic organisms. I know that some of the big blooms that have happened off of the eastern seaboard, and I think actually a little bit in some places in I think Oregon, they can actually off-gas a toxin that can blow in as this like aerial cloud that's particularly nasty. And so that's one kind of out of the sea itself effect that you can have from certain species.
The ocean's such a fascinating place.
Mm-hmm.
So this is Nicole just signing off the Q&A for Nautilus Live. Thank you to everyone who sent in their questions, and we appreciate you following along. Sorry we couldn't answer them all, but we'll plan to do this again. Great to get feedback from everyone.
Thanks for reading them, Nicole. It was fun. Little deep sea soul at the bottom of the screen. Lots of these umbellula. I'm used to seeing umbellula around, but I don't think I've ever seen so many in one area. They're definitely the most numerous coral around right here. And tanner crab.
Meredith, I'm noticing the oxygen saturation seems to be a little bit lower than when we started our shift. What's a normal range of oxygen saturation at the seafloor? Or is there, I know it depends on which ocean you're in and a bunch of other things, but what do you think about that?
Oh, I cannot remember off the top of my head, actually. I know it's definitely, it is lower than you would see at the surface waters, but it can be quite variable depending on what water body you're in. I would have to look up what we would consider to be typical for this area.
Just thinking about hypoxia and how often in August and September, I think especially off the coast of Washington, we've seen some hypoxic events in the past. I'm wondering what the threshold is for that. I'm sure we're way above it right now, 0.6 kind of range, but just wondering. Well, it looks like we have a problem with the O2 sensor, so I don't know if that's actually accurate.
I want to say it's 2 parts per million is the hypoxic threshold. But here it's hard to say. I'm not used to looking at the numbers in the oxygen saturation, and I think we'll be checking in the calibration, but you do expect this deep kind of a variation in the oxygen. There are some areas that can be quite low just naturally, this deep in the ocean, and part of that's the ongoing bacterial activity has just used up what's available, and because you're not mixing down from the surface, it doesn't get replenished that quickly. That's where you get things like long-term oxygen minimum zones. So if we see a low oxygen, it will just depend on what the other oceanographic conditions are, whether that's natural or whether that's more of an event.
So just a quick question. Do you anticipate a lot of sampling in this last hour and a half that we're down here?
It depends on what we see. Right now with this mud, the only thing I can think we might take if it's the right thing would be one of the sea pens.
Okay.
Or maybe one of these. That's a stocked sponge there on the bottom, but that's one we've collected on previous cruises. But I don't think there will be a lot unless we get into some more hard substrate.
Okay, I'm just making some pretty aggressive steps to cover ground, so the more heads up you can give us, the faster the better, then I could stop the ship.
Yeah.
Thank you.
Yeah. So the sponge that we just went off the bottom of the screen there, and we might get another one up here. We call it a fiber optic sponge sometimes because they grow these long spicules that they use to attach to the soft substrate on the seafloor. And if you get one of these up to the surface, it actually looks like a wound up fiber optic cable. They're very, very tough.
Yeah, I'd like to join the oxygen conversation for a second. This is an area that very naturally has low oxygen, and that's because it's some of the oldest water in the world since it was last in contact with the atmosphere. So a lot of the water that is off the Pacific coast, and especially at the depths that we are here, which is in the oxygen minimum zone, the last time it met the atmosphere was in the North Atlantic, and it's been in the deep sea ever since. So that long time is why it's nutrient rich, and when it comes to the surface, it fuels the highly productive coasts that we have off Oregon, Washington, and California.
Yep, we're an upwelling zone, so the wind that comes off the ocean and combined with the current actually serves to transport this deep water up towards the surface. And that's one of the reasons we're so productive. The eel pout there behind one of these fiber optic sponges.
Did I hear a request? Sorry.
Oh no, I was just narrating what those species we just went over are.
Oh, sorry. I think I heard an internal control van. Sorry. Never mind. Thank you.
Oh. Couple more thorny heads. See, when I saw the first one of these sponges, and we have another one coming up here, it's hard to recognize what it was because there were so many crinoids growing over the stalk, or climbing over the stalk. This one's pretty low to the mud.
Is the sponge this guy coming into lasers here?
Mm-hmm.
Interesting.
I think that's a sponge. If you have
Like a
time, we can
candle.
quickly zoom on it.